Welcome to the next unit, and in this unit, you will learn everything you need to know about cellular energetics.
The guided question I want you to keep in mind today is how do competitive and non-competitive inhibitors affect the reaction rate? Okay, so turning reactants into products. And this is the chemical reaction, and enzymes would speed that up. So how do these inhibitors affect this reaction rate? Now we get two types. We get competitive inhibition, meaning that the competitive inhibitor and the substrate are competing for the active site. Whereas non-competitive inhibition means that they're not competing at all, that this non-competitive inhibitor binds to a different site on an enzyme that changes the active site so that the substrate is not able to bind. So they're not competing at all. Now, allosteric enzymes. Okay, allosteric enzymes. These enzymes get their name from allo means in a Greek origin, it means other, and steric means shape. So it's enzymes that have another shape. Okay, and they can also change their shape based on this site it has. So enzymes that we've already learned, they have an active site. And this is the binding site for these substrates. Okay. Now, some enzymes also have other shapes, and they have another space that is called the allosteric site. Now, this is where a different substrate can bind, and once it binds, it changes the active site so that the active site becomes inactive. It can do either two things. It can bind, and then it can activate the active site, or it can deactivate, meaning that it can either increase or decrease the activity of that enzyme. For competitive inhibition, okay, because they are competing for the same active site, okay, and it does not change the 3D structure. So the 3D structure is the same. They are just competing for this active site, okay? Now, in normal enzyme activity, if I increase the substrate concentration, the reaction rate will increase up until a point until all the enzymes are saturated. Because now the amount of enzymes is a limiting factor. Okay? Now, what happens with competitive inhibition? Because they're competing for the same site, it will bind to the active site, but it will not change the shape of the enzyme. But because it has bound to the active site, the substrate cannot bind. And if the substrate cannot bind, it cannot catalyze the reaction. So the reaction rate will go down. Okay. So here we can see, even in a low substrate concentration, when it's normal, okay, the reaction rate is higher. But if I add a competitive inhibitor, okay, the reaction rate will be lower because it's competing for that active site. Now, what is great about competitive inhibition, okay, because it affects the substrate binding at the active site, if I keep on increasing the substrate concentration to have a very high substrate concentration, meaning that I add even more substrate than there is inhibitors present, then it's more likely that it would reduce the inhibitor's effect. Okay, so at a very, very high concentration of substrates, it will almost be the same or just a little bit lower than the normal enzyme function, which means that if you want to overcome competitive inhibition, you just add more substrates. Non-competitive works a little differently because in non-competitive, we change the 3D shape. Okay, so we actually change the shape of the active site. So once an inhibitor binds to that enzyme, the active site changes, and that means that this enzyme is no longer able to bind to the active site. Okay. Now, the difference here is if I add more substrate, it does not restore the reaction rate because the enzyme's activity has changed. So here I can see, even if I start adding more substrate, the active site has changed. So it's unable to bind to the active site in the first place. Okay, so here let's do the normal enzyme first. The reaction rate will be very high. And at the same concentration, the reaction rate will be very low for non-competitive inhibitors. But even if I start to add more substrates, it would still stay very low, okay, compared to normal enzyme activity that's very high, because it's not competing for the same active site, and it has changed the 3D shape of that enzyme by binding to this allosteric site. If you like that lesson, don't forget to like and subscribe, and I'll see you for the next one.